Now the composition of the human body is almost similar to all the mammalian vertebrates, similar, and formed by the elements, different elements, and composition as in compounds, and there are some values you can remember regarding the elementary composition. Most is carbon, 50% in weight basis. In weight basis, 50% is carbon. Then oxygen, it is 20%. Hydrogen, 10%. Then nitrogen, 8.5%. Calcium, 4%. Phosphorus, 2.5%. Potassium, 1%. They altogether do not form 100%. In spite of that, I can remember these values. It is taken from a biochemistry text of older editions. Yeah. This is by the weight. This is by weight. This type of composition. And there are also other elements in decimal fraction. Decimal fraction. Like sulfur, 0.4%. Sodium, 0.8%. Excuse me, 0.4%. Chloride, 0.4%. And others are, suppose, others are even less than this. Suppose in case of iron, it is 0.01%. And I cannot remember everything in details because of my poor memory. I cannot remember everything in details, but it will give some idea how it is possible to form a body, not only the human body, but also other mammalian vertebrates have almost similar composition. Panchabhute deho toiri hoy, and when a man or animal is burnt or buried under the earth, then they decompose and they will mix. So this is the body, structural composition. There is hardware, and what is moving us, there is the ego or I, and there is the software. And it bears some energy system. You have learned in your physical chemistry or physics, energy system, closed energy system, open energy system, and this type of systems. Isolated energy system, and all the living things are open energy system. They can take mass and energy from outside and liberate this. You should, when you enter, you should go to the nearest post. It is the old habit of all the students everywhere. Whenever this type of system were not there, public address system, and then the students have to go to the rearest position, because majority of the students, they do not want to see, want to listen what we have talked. It is not a musical sound. And so they try to avoid. And they are intelligent enough how to avoid, because the intensity of sound is inversely proportional to the distance produced to the sound producer. So they go rear and rear. বুঝতে পারিস? এগুলো যে প্রাচীন যে অভ্যাস সেটারই পরিণতি। Unfortunately, you cannot avoid my sound wherever you go. So you have to go to the nearest post, so that energy will be less consumed. বোঝা গেল? এই জন্য বাংলায় বলা যায় যে এটা হচ্ছে যে ইনভার্সলি প্রপোর্শন, ব্যস্তানুপাতে রয়েছে, বর্গের ব্যস্তানুপাতে রয়েছে। এই জন্য যত দূরে যাব তত শব্দটা কম যাবে, এবং এই শব্দ কানে যেন না যায় সেই চেষ্টা করতে হবে। And the ear has the physiology of regarding this, and we can discuss whenever we learn the hearing process. There are neural tract by means of which we can avoid sounds. আমি শুনবো না, বুঝতে পেরেছি? যেমন গভীর নাস্তিকরা আমি কিছুই ঈশ্বরের নাম শুনবো না, সে যখন চেষ্টা করে কিছুটা সফল হয়, and that is a part of physiology. Everything good and bad is included in the subject of physiology. So those are bad people like me, they have learned everything and they know something of this every detail, in good and on the bad sense. Now this is the elementary composition. 50% is carbon, and you see the coal, it was once a life trees, and they are providing our energy and our food. Oxygen, hydrogen, nitrogen, calcium, phosphorus, and then the potassium, and these are this, and in this, and also iodine also in this included, 0.00045, and this is this. So you have to go through books, old texts of biochemistry, where there it is given, Harper's biochemistry. Now in compound, but it is in the weight basis. In the weight basis, bears less value, because they are present as molecules. Equivalent molecules are more important when you consider its metabolic activity also, how they react. And in compounds, then it is asked in the examination. It is not asked in the examination. It is for only understanding. In the suppose in an individual 60 to 65 kg, 65 kg or 60 kg in the individual, and in that case, and you take the condition compounds, then water is the majority part. Water, it is 40%. Excuse me, 60% is the water. 60% is water. Suppose protein 17 to 18%, 18%. Carbohydrate 1%, minerals 7%, and the rest is fat. You can find out. These are the average values. It will vary. Now this body is formed of these compounds. Water 60%. It is divided into two parts, two compartments: extracellular fluid and intracellular fluid. There are billions of cells in our body, and roughly 1/3 is extracellular, 1/3 ECF, and 2/3 ICF. And these are this value. In Bengali, extracellular fluid, কোষ বহির্ভূত তর, আর ICF is কোষ মধ্যস্থ তর. So this is the composition. And this body, when you consider as cellular structures, cellular structure is the unit, is the cell, numerous similar cells or even dissimilar cells combine to form a tissue. More than different types of tissue combine to form an organ. And similar type of organs, they form the organ system, like musculoskeletal system, gastrointestinal system, genitourinary system or system, and respiratory system, cardiovascular system, you come in. Now, so we should go at first to the cell unit, a cell. And there is no cell is typical. No cell is typical. Every cell is, they have some common characters and also some unique properties. Suppose in case of RBC, it is also a cell. RBC. It is also a cell. And hepatocyte, liver cell. And it is also a cell. Lots of differences. According to the function, the body produces this. RBC does not require any nucleus, but this has prominent nucleus and protein synthesizing machineries. So when up to describe in the text, they describe the liver cell. It contains almost all the organelles. RBC size is suppose 7 to 8 μm. The liver cell, it is to some extent more than this, and it contains nucleus and all other organelles. And the cell is surrounded by a membrane, and it is called plasma membrane. And its primary idea is to isolate the cell from the watery part. Life originated in the sea, and water everywhere. And how to separate some component with it? That is water insoluble. তেল জলে মিশ খায় না, এজন্য a lipid structure outside. This is the nature's device. কি করে সে আলাদা কম্পার্টমেন্ট তৈরি করবে, বুঝতে পারছেন তো? নেচারের ডিফারেন্স আছে বলেই তো এতগুলো পলিটিক্যাল কান্ট্রি তৈরি হয়েছে আর পরস্পর বিরোধের সৃষ্টি হচ্ছে। And it is the nature's device, so that it can isolate its contents from the surroundings. So lipid must be there. Otherwise, how it is possible? Yeah. And phospholipid is the phospholipid. Similarly, within the liver cell, there are other organelles like this type of organelles. So they are separated by membrane. It is called unit membrane, plasma membrane, formed by made of the phospholipids. Phospholipids you will learn in detail in biochemistry, and in higher secondary course you have also learned to some extent. Majority is the glycerophospholipids. Glycerol moiety. Suppose this is the backbone of glycerol. Fatty acid 1, fatty acid 2 in this position, and this position is a phosphoric acid moiety, and it will form the phosphatidic acid. Then with some base or this type of structure can unite with it. It may be choline, it may be ethanolamine, it may be inositol in this. It is the phospholipid, it is the phosphatidic acid, this part. And this remaining part, it may be choline, it may be ethanolamine, it may be inositol or other substance, phospholipids. And you see where there is phosphate group, and it is the polar group. it is the polar group. It can bind with, that is the watery part, hydrophilic part. And so you can consider this part as the hydrophobic part, and this part is the hydrophilic part. Hydrophilic. That's two components. And the hydrophilic part will try to make contact with water, and water is present in the extracellular fluid and in the intracellular fluid, that is called cytosol. And this hydrophobic part, they avoid water, so they will aggregate together. So instead of one layer, two layers are better in this case. In the energy, this energy based position, and it is the best position. Clear? Suppose you see the water falling during rain, and it is a spherical structure, because the surface area is minimum. So it is due to this energy like matter. So it is double layer, two, there is lamella, singular lamella and lamellae, and it is distributed in this way. Suppose and in this way they are disposed. It is the hydrophilic part, it is the extracellular fluid, and it is the membrane. Average dimension is 7.5 nanometer. That is 75 angstrom in it. Nanometer. 10 to the power minus 9 meter. Nanometer. And it is this phosphate containing part, and it is the, and in this case it is the cytosol. It is this, and in this way they are disposed. Double line. And there are some spaces in between these molecules, and the phosphorus, and some prefer this external lamella, some prefer this internal lamella. Like phosphatidylcholine, ethanolamine, there is, and they prefer the external lamina. More details you can have in biochemistry, we are not much more interested. Now within this, and there are these other lipids also, other lipid like structure like glycolipids, sphingolipid, they are sphingomyelins, they are. Clear? They are sphingosine derivatives, and also cholesterol. And these fatty acids are not similar, they are dissimilar. And in the two position, stereospecific numbering, it is this, they are, and in this case, they are unsaturated. In some cases, polyunsaturated, and from which prostaglandins can be produced. Polyunsaturated fatty acid. Yeah. But in the one position, they are relatively, there is saturated fatty acid. And other lipids are sphingolipids or sphingomyelins or this, and also cholesterol. Sphingolipids are produced in that case an amino alcohol, sphingosine, derived from palmitic acid and serine, derived from sphingosine, amino alcohol, and from which this they are derived. Sphingosine. structure formula, and it is the idea so that you can compare this within the book and you try to find defect. Then it will make you clearer. Yeah. This type of it is sphingosine. And when a fatty acid unites with it, then it becomes ceramide. Now, in this case, inositol can combine. Inositol may contain some carbohydrate moiety, and to which the protein can unite in this compound. So this type of compounds are there. And in this way they form this glycolipid, and there is and in this case there may be some combination of suppose choline, and in that case they form this sphingomyelin. I am requesting you today you must open the biochemistry text and go to the structure of the lipids and sphingomyelins and sphingolipids, then you will have idea. And if I have committed some mistakes, you will explore it, and then you will be happy that our knowledge is perfect. The class was an imperfect one. In spite of that, we should go progress so that your knowledge is the concept, not my knowledge. Clear? And in this way, there are also cholesterol. And when they are present, then this membrane structure will change, and this fluidity will increase in some cases. Now, by weight basis, that is this lipids and the proteins, and proteins are embedded into it. It may be just touching it, or it may be embedded in the bilayer. So there are two types of proteins, peripheral proteins and integral proteins. Suppose if there is some membrane, some proteins that are just touching, it is the membrane, and it is just touching this lipid, it may be touching this some lipid, it is the peripheral protein. And if it is, and this type of proteins are peripheral proteins, and some going to the part of it or even through and through. There is the integral proteins. And the amount of proteins and the lipids, they do vary according to the requirement of the system. On the average, 50% of the body weight, this is the weight of this membrane is protein, 50% is lipid. But proteins are larger molecules, and their molecular weight is much more than this. So whenever consider the molecular basis, one molecule of protein is equivalent to 50 fatty acid moieties, 50 phospholipid moieties. This is the gross average and varies in cell to cell. Yeah. This is the peripheral proteins, and these are the integral proteins. And some textbooks, they complicate the system. They are called extrinsic protein or intrinsic protein. It is difficult to grasp and bears nothing more than academic value. Now they are floating in this mosaic position, fluid mosaic model, mosaic separated structures floating in this. Now these proteins have numerous functions. And how these peripheral proteins attach, it depends upon this character and this ultimately the product that is produced. Peripheral proteins, how they are, some may unite with some, that is the fatty acid moiety protein. Some may form ester form and some other forms in which they can combine. Suppose in case of, you consider the case of suppose you draw a line, one is the lipid soluble and another is the water soluble part. And whenever there is some, suppose in this case, myristoylated part, myristic acid, CH3, CH2, 12 COH, and it bond with the, that is the N-terminal part of the protein. Or in some cases, it may be in other ways. Suppose in case of palmitic acid, it is the N-myristic acid, palmitic acid, CH3. In this way, it can unite. And in this way they are placed. And in this passage, long chains, they are the hydrophobic part, and it is the hydrophilic part in that case. NH2 group and other groups where it is water-loving hydrophilic group. Or in there may be some geranyl or farnesyl groups in some cases. They can unite in this way. In your chemistry, high secondary chemistry text, you will find this type of compound. Isoprenyl group. Suppose it is rest is like this. Geranyl group, and here additional CH3 group. Geranyl group. Or if it is 2, then it will be farnesyl group. And these are all lipids, water insoluble structures. It cannot bind with water. They are not polar group, they have no polar groups. Water itself is to some extent a polar compound. Within the molecule there is some excess or deficit of electrons in this water molecule, and that is responsible for some unique characters of water. So in this way they are disposed. When it is 3, it is geranyl, or it is 2, it is farnesyl group. You read the book and you will find this and try to find defect, my defect, and it will be beneficial for. And there are also GPI anchors, glycosyl phosphatidyl inositol linked proteins, GPI anchors. And in that case, GPI anchors, in that case, that is the cyclohexane. It may bear some monosaccharide group also, some carbohydrate moiety, and with this, by means of another phosphate, it can unite with the protein. GPI anchors. It is called GPI. GPI anchors. Numerous proteins are combined in this way. Glycosyl phosphate, GPI, it is the abbreviation you will find in the text, and in this way proteins can be attached. It includes the alkaline phosphatase. There are some factors, that is the protect the cell from the complement lysis, and also some antigens and other proteins, 40 proteins, I cannot remember everything in detail. And in the GPI anchors are also there. GPI anchors are there. Some drugs can unite with it. Glycosyl phosphatidyl inositol linked these proteins. They are in this way. For clear, accurate value, you have to, this structure, you have to consult the biochemistry text. In this way, they are produced. So, in this way, the peripheral proteins, they are united on the outside and also inside. And there are also multiple proteins, they are through and through proteins or integral proteins. And they are produced within the cell. And they are targeted, they are by sophisticated means. They are produced within the cells and they are targeted. In this way, there is this cell, basic structure of the cell is produced, and the function of the plasma proteins are important. Plasma proteins are important. Water and small molecules, even if polar or non-polar, they can enter it through the membrane. In between there is these phospholipid molecules. Through the interstices they can pass. Water, carbon dioxide, oxygen, they can pass. But not other structures which are surrounded by water molecules. Water molecules, they cannot enter to the membrane easily. So they require some transport proteins. Here, sodium ion is surrounded by water molecule, and the hydrated form of the sodium ion have larger diameter than that of the potassium ion. Atomic weight of potassium is more than sodium, but hydrated form is relatively lower. And there are some channels. There are some channels within this membrane that can permit the potassium. But sodium channels are relatively less in number, and they becomes activated when it is required. So important function, and there is the semi-permeable membrane. Every cellular unit membrane is a semi-permeable membrane. Yeah. It will allow some entities to enter and do not allow others. যেমন দেখো না, আমাদের এখানে যারা বড় 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 মানুষ দেখবে তাদের সঙ্গে সহজেই অনেকে যোগাযোগ করতে পারে, বিনা অনুমতিতেই চলে যেতে পারে। হ্যাঁ, কিন্তু আমাদের মতন যারা লোক মানে অবাঞ্ছিত লোক তারা এখানে ঢুকতে পারে না, অনেক দূরে পুলিশের লাঠি খেয়েই অনেকে ফিরে আসে, বুঝতে পেরেছি? সেরকম এভরিথিং ইজ গোয়িং ইন দি সেম ওয়ে ইন দি নেচার। সেমি পারমিয়েবল মেমব্রেন, potassium permeability is more than the sodium permeability. And most of the water soluble molecules, they have some water coating and their effective molecular size increases and they cannot easily enter. And so they require different type of protein channels, and the protein channels are formed of subunits, so that they can allow some substance to go through, and they are preferential channels. Some are sodium channels, some are potassium channels, even independent of the sizes, and they are due to the amino acid composition of the lining of these channels. And in that way they can allow some molecules to enter preferentially. Yeah. And some of the protein molecules are cell adhesion molecules, CAM, cell adhesion molecules. Cell adhesion molecules. Suppose 20 years ago, the term is rarely found in the text, common text, but it is coming into every text. Cell adhesion molecules, by means of which cells are attached to one another and also with the basement membrane material, and they are of importance, but because they are required for the proper development, fastening of the cells, structural cells, and also they have some effects similar to that of the hormones. Information can be transmitted through them, and they are involved in inflammation and even migration of the cells, during propagation of these cells, malignancy. So they have importance, and they are of different groups, cell adhesion molecules, and some proteins, plasma proteins are pumps, wave-like pumps. They may include enzymes. Whenever the flow is following the electrochemical gradient, then it is a passive process. Diffusion is a passive process. বোঝা গেল? যেমন যদি ফ্যান নাও চলে, ঘরের একটা কোনায় যদি একটা ধূপকাঠি জ্বালানো যায়, খানিকক্ষণ পরে সেই গন্ধটা আমাদের আসে, যদি মুভমেন্ট নাও থাকে, অ্যাপারেন্ট মুভমেন্ট নাও থাকে, সেটা হচ্ছে ডিফিউশন। ইট ইজ দি বিহেভিয়ার অফ দি মলিকুলস টু মুভ randomly from high concentration to the low concentration, and from the low concentration to the high concentration, but the net movement from the high concentration to low concentration in the process of diffusion, it depends upon the concentration difference and the distance or the depth and its nature. Some can diffuse easily in comparison to others, and in case of membrane, it depends upon the solubility of this. Now, when it occurs, it is called passive motion. Passive. Yeah. Passive. Suppose within the alveoli, oxygen concentration is more than in the pulmonary capillaries, because blood is draining from the lungs to the arteries to the tissues, and tissues are utilizing oxygen and replacing it with carbon dioxide. So when it returns by the pulmonary veins, go to the right side of the heart, then pulmonary trunk arises and it branches. Ultimately, pulmonary capillaries are going into the alveoli. Within the alveoli, the concentration and partial pressure is more, so that oxygen is diffusing through this. Diffusion is physical process. No cellular energy is required. Yeah. Similarly, carbon dioxide is present within the alveoli, but in low concentration than in the pulmonary capillaries. So in the reverse direction, carbon dioxide diffuses. Carbon dioxide is not present in the atmosphere. Negligible amount present. Carbon dioxide is produced within the body. So it has to be eliminated. And respiratory system ventilation is primarily for the carbon dioxide elimination. So the concentration of carbon dioxide in the pulmonary capillaries is more than that of the alveoli. And from the alveoli, it is more than the atmosphere. Clear? 45-40-0. And within the alveoli, and it is a dynamic equilibrium. And so carbon dioxide in the reverse direction. And it is a physical process. No cellular energy is required. In the adult individual, we are utilizing in the normal condition 250 ml of oxygen per minute, and we are liberating 200 ml of carbon dioxide per minute, and having the respiratory exchange ratio of 0.8, 200 divided by 250. Yeah. And it is going on. No cellular process is required. Nature is 10% intelligent in that sense. And in that case, no cellular energy is required. Cellular energy is required to cause the ventilation. Little energy is required, ventilation. Otherwise, this gaseous exchange is passive process. But in some cases, it is not adequate. Though it is passive, it is not adequate. Suppose glucose is entering from the extracellular fluid into the cells, and within the cells, glucose is being constantly consumed. So its concentration is low. So glucose from the exterior can enter into the cell by the passive process, but it is very slow, because glucose is a large molecule and surrounded by also water. So it requires some transporters. They are called carriers. Glucose transport, GLUT4, GLUT2, they are, and they are required. And this diffusion is called facilitated diffusion. ব্যাপন প্রক্রিয়া বলে বাংলায়, ব্যাপন প্রক্রিয়া। আর এটা হচ্ছে সুকৃত ব্যাপন। বুঝতে পেরেছো? বিদেশি ভাষার সঙ্গে সঙ্গে নিজেরটাও জানা থাকা দরকার। হ্যাঁ, যে সমস্ত লোকেরা আমাদের মধ্যে মূর্খ, তারা নিজেদেরটা আগে শিখে নেয়, তারপরে অপরের দিকে তাকায়। বুঝতে পেরেছো? They do not suffer from any inferiority complex. They try to learn the language in their mother tongue, and they can then explain in any way. And this knowledge becomes more deep and permanent. মাতৃভাষা শিখলে খুব সহজেই সেটা বোঝা যায়, অল্প সময়ের মধ্যে শেখা যায়। কালক্রমে এরকম হবে যে বাংলাতেই তোমরা, হিন্দিতে অলরেডি চেষ্টা হয়েছে যে মেডিকেল এডুকেশনটা মাতৃভাষাতেই এখানে হওয়ার কথা, কিন্তু পশ্চিমবঙ্গে সেটার সম্ভাবনা খুব ক্ষীণ, কারণ এটা একটা খুব ডেঞ্জারাস পজিশনে আছে, কাজেই এখানে সেটা হবে না। সো উই হ্যাভ টু গো থ্রু দি টেক্সট ইন দি ইংলিশ। এবং অনেক টিচার আছে, তারা বাংলায় উত্তর দিলে সেটা সত্য হলেও মেনে নিতে চায় না। বুঝতে পেরেছো? দীর্ঘদিন দাসত্বের ফলে এরকম হয়, তার ফলে এরকম মেনে নিতে চাইছে না। আমি তো ঠিকই তো বলছি, না ওইটা ইংরেজিতে বলতে হবে, ইংরেজিতে বললে তবে সে সন্তুষ্ট হয়। বুঝতে পেরেছো? সেই জন্য তোমাদের ইংরেজিটা তোমাদের এসেনশিয়াল হয়ে পড়েছে। আর আমি যদি ক্লাসে বাংলায় বলি, তখন আমরা বলবো গিয়ে বাংলায় বলছি, আমরা বুঝতে পারছি না, এরকম। So these are this. So and pumps are wherever it is against concentration gradient and electrical gradient. Suppose in every cell, every living cell, that is the inside is negative in comparison to outside. So, but the concentration of sodium and potassium is such that it requires some pumping effect. Intracellular sodium is in low concentration than extracellular fluid. In the extracellular fluid, it is 140 to 150 millimole per liter sodium concentration. And within the cells, suppose it is 10 or 15 millimole per liter. So, for passing sodium from inside to outside is required, and it requires some pushing system, cellular energy system. So, some pump is there, and these are also channels, and they behave in such a way that there is generation of energy. ATP is broken down. So, it is the ATPase, sodium, potassium, ATPase. Yeah. Potassium inside is much more than the outside. And so, from outside to inside, entry of the potassium is difficult. It is not possible by the process of diffusion, even by facilitated diffusion. And there must be a sodium potassium ATPase is there. It is present in all the living cells membrane, and it is its marker. There are some elements in the membrane that are called markers, and it is present. And the different membranes, unit membranes within the organelles, they may have different markers. Suppose peroxisome, it has a membrane, and there are some elements that are peculiar to that organ. It may be catalase, uric acid oxidase, and there is the markers. If any membrane have this, then you are sure that it is coming from this membrane, is not from any other cell. It is coming from, it is peroxisome, markers. Yeah. And also other organelles or some protein molecules or larger molecules, they have some markers. Now the enzymes and also carriers. In some carriers, suppose glucose transporter, they are called carriers. They transport one substance following the electrical and constitutional gradient into this. They are called carriers. Some other membrane proteins are receptors that can bind with unique agents, hormone receptors, and they have some specificities. Some may be within the cells, nuclear receptors. Some are outside the cells, and they have different, and there are similarities and dissimilarities in case of the hormone. They are called hormone receptors. And when you unite to the appropriate receptor, then it can have its function. And the cells differ in their presence of the receptors. All cells do not have the receptors of all the hormones. Typical examples, suppose in case of glucagon, the liver cell has its receptor. So glucagon has its effect upon the liver cells, and primary function is glycogenolysis. But the skeletal muscle do not have the glucagon receptors, so it is ineffective in that sense. But epinephrine has receptors both in the liver cells and also in the muscles. So epinephrine can have effect on both of these. From the liver cells, it can generate glucose, but in the muscle cells, it cannot generate glucose. It can form lactic acid. That can be reconverted by neoglucogenesis into the liver and may be converted into glycogen. So the receptors, and there are some proteins about the cells that is the antigen recognition and antibody processing. Blood group substances are there. They are in the cell membrane, but we differ. Our blood are red, and due to this hemoglobin. But there are groups. There is the plasma membrane proteins, and we are not same in this chemically also. Only anatomically there is some similarity, and biologically and mentally we are all different. By DNA fingerprinting and by this fingerprinting of the fingers, and it is evident we are all different. Some are O group, some are A group, some are AB group, some B group. So that, and it is a function of the membrane proteins and antibody processing, antigen recognition and antibody processing. Details you will have in the discussion in the immunity, in hematology classes, and you will also read, and you will make habit to read yourself and know yourself. Everywhere what you see is not exactly what you think. তোমার দেখা আছে যে এখানে একটা বিভাগ আছে, এই বিভাগে সমস্ত কিছু শেখানো হবে বলে তোমার অনুমান হলো, কিন্তু সেই অনুমানটা প্রায় সত্য হয় না। বুঝা গেল? এটা বাংলায় বললে বুঝতে পারো, অনুমানটা সত্য হয় না। গিয়ে দেখবে অমুক দিন ক্লাস হচ্ছে না, অমুক দিন অমুক বাধা, কোনোদিন ছাত্রদের কোনো আন্দোলন হচ্ছে, হ্যাঁ, সবই মূর্খামির মানে রিপ্লেস। এখন এই জন্য হচ্ছে কি তোমাদের নিজেদের জানতে হবে যে তোমরা ইউ হ্যাভ টু মেক ইউর হ্যাবিট টু লার্ন ইয়োরসেলফ। কে তোমাকে শেখাবে, তোমাদের সেই দিন পার হয়ে গেছে। নাও ইউ আর অল অ্যাডাল্টস, এবং তোমরা হয়তো ভোটারও পরিণত হয়েছে, জ্ঞান বুদ্ধি তোমাদের অনেক প্রসারিত হয়েছে। সেজন্য কোনো ক্লাস হয়নি স্যার, পড়ানো হয়নি স্যার, ওই সমস্ত বলার দিন চলে গেছে। তোমরা নিজেরা এখন পড়ে নেবে এরকম আশা করা যায়। যখন যেটা ক্লাসে যখন বাকি থাকবে যে এই কম্পিটেন্সি সবগুলো হলো না, তখন আমরা তোমাদের নোটিশ দিয়ে দেবো, ইউ হ্যাভ টু লার্ন ইয়োরসেলফ। ভিন্ন ভিন্ন টেক্সট বইয়ে লেখা আছে, টেক্সট বই ইন্টারনেট থেকে, আর তোমাদের উন্নততর এবং আরো অধিক মেধার লোকেরা তোমরা এসছো পৃথিবীতে, এবং তোমরা সেটা শিখতে পারো। এমন কখনো বলা চলবে না যে ওটা ইয়ে হয়েছে। প্র্যাকটিক্যালের সম্বন্ধে বলতে পারো, স্যার প্র্যাকটিক্যালটা আমাদের ওটা হয়নি, সেটা আমাদের করা যেত। কাজে সিলেবাস সবসময় শেষ হবে কিনা এমন কথা বলা যায় না, কিন্তু তোমাদের এটা নিজের অভ্যাস করতে হবে, নিজে পড়ে ভিন্ন ভিন্ন বই। হ্যাঁ, এটা তোমাদের অভ্যাস করতে হবে। কখনো আশা করবে না সবসময় কেউ থাকবে তোমাদের কাছে, সমস্ত জিনিসটাই পড়ানো হবে এমন কথা বলা যায় না। উচিত অনেক কিছুই, কিন্তু সেটা বাস্তবে হয়ে ওঠে না। These are the functions of this. Now this cell adhesion molecules, it has also different groups like selectins, integrins, immunoglobulin G superfamily, cadherins, four groups: selectins, integrins, IgG superfamily, and cadherins. Four groups. Selectins are sugar binding proteins, complex sugars. They can unite with the complex sugars. Lectin binding, there is a term lectin that is present in some molecules that can bind with sugars. Containing other part. It may be intercellular some molecules, or even in the other cells it can bind with particular sugars, and it contains different type of motifs. Charlie-Lewis motif, and they unite with any element present in another cell or in the interstitial fluid that can unite with them. That selectins are of these varieties. Different types of selectins, and they are present upon the different types of cells like neutrophils, lymphocytes, and their number may increase in some cases. Selectins, they are responsible for the rolling of the neutrophils over the epithelium, initial part, and then its binding and emigration. This type of, that is the cell adhesion molecules are there. And this integrins, they are heterodimers, alpha, beta in this, heterodimers, integrins. And they can unite with the different molecules, cytoskeletal molecules, and also other parts within the cell and also outside. Integrins, heterodimers, And when this same type of molecule unite, it is called homophilic binding. And when it is not, it is called heterophilic binding. Suppose when these integrins unite with the IgG superfamily, like ICAM also, intercellular adhesion molecule, then it is heterophilic binding. And when one integrin unites with the other integrins, then it is homophilic binding. Or when cadherins, calcium binding adherent proteins, it can unite with another calcium time, and that is called homophilic binding also. These are called, there is the cell addition molecules. These are 4 groups: selectins, integrins, immunoglobulin G superfamily of proteins, and also cadherins. And they unite cell to cell and even cell to the extracellular material, elements of the basement membrane also. And they have some importance. And some of they have send information into and outside of the cell in conditions. And they are responsible during development and even the spread of these tumor cells. When the malignant cells, they develop, and their adhesion molecules may change in such a way that they can ease of transport to another cells, either local spread or through the blood vessels. In inflammation, they have role. Neutrophils come outside the cells, outside its lumen, and when they accumulate, they form the pus-like structures, neutrophils, and it is due to their selectin integrins and the ICAM of the interstitial cells. Cell addition molecules, these are. And within the cells, there are the cytoskeleton. In individual, there are the skeletons. What is the function of the skeleton? It is maintaining the shape and size, and also it is associated with the movement as a whole and part of it. Similarly, within the cell, there are cytoskeleton. They are different fiber-like proteins. And along with this, there are also molecular motors, cytoskeleton. And cytoskeleton, there are these fibers, there is microfilaments, microtubules, intermediate filaments. And they form structural basis mainly, and also they assist in the movement with the help of molecular motors. Molecular motors are different types. They may be myosin, they may be dynein, they may be kinesin, and in some cases they may be dynamine, endocytosis. And time is limited, so I am leaving this. Next day we will proceed in this way, and I will try to complete this part in the general physiology within the scheduled time, and if not possible, then you have to learn yourself. Self-learning you must practice. You should not depend on us, because we are not reliable, completely reliable. Yeah.